Okay, good afternoon, everyone. I hope that you are doing well on this chilly day. Let's see here. Do you have any concerns or questions about the course, about our schedule, about anything moving forward? You know, we are about 3.5 weeks from the end of our semester, so things are really going to start feeling accelerated in the sense that things are going to start wrapping up and all of a sudden we're right at the final exam. So do you have any questions at this time that I might help you with? Okay. If you can hear me okay, please type yes into the chat window. Okay. Thank you very much. All right, gang. Then let me just remind you that your second literature review is due this coming Tuesday. So again, please type your answers into the Microsoft Word document that I sent along with the article. And please email me that no later than the end of the day on Tuesday of next week so that I can evaluate those and get them back to you. Okay, so we're going to jump back into chapter 11, try to finish that up, and then get into chapter 8 after that. And I'm going to try to get through as much of chapter 8 as I can, including the possibility of doing a little bit of extra lecture with you in the office hour slash lab virtual hour that we have on Thursday. So let's begin, gang. I'm going to go back to Microsoft Word document here. Okay. And certainly if you need my attention, please feel free to type into the chat window. Okay. At the end of last class, we were talking, well, we had just finished up a pretty extensive coverage of a variety of things that can happen in the environment to actually damage our DNA, to create DNA mutation. And we just got started the general topic of DNA repair systems. So as was the case last time, this is going to be kind of a show and tell section of the lecture where I make a statement or two and then show you a video about various repair mechanisms that cells can actually employ. Okay, so the first one that we covered at the very end of last class was the fact that DNA polymerase enzymes can often repair mistakes in the DNA because they have this 3' to 5' exonuclease activity that allows them to go in reverse. A second and new thing for your notes is something called mismatch repair. Okay. And in this case, what happens is that enzymes are able to identify and repair a mistake in one strand of the DNA, in part because DNA that's in the cell, the longer it's in the cell, the more likely it is to have epigenetic tags, which means cells can kind of tell the difference between old strands of DNA that have been around for a while and new strands of DNA that haven't been around for a while based on their methylation status. So if a potential error is detected in a new piece of DNA, as judged by the absence of epigenetic tags, then the repair machinery of the cell can go ahead and fix it using the older, more mature strand of DNA as a template to fix the error. Okay, so let's see here. In this scenario, cells use enzymes to detect the relative age of strands of DNA based on epigenetic tags. Older DNA strands will have more epigenetic tags than younger DNA strands. Okay. All right. So gang, I want to show you a video based on this, and hopefully the video will speak for itself. So bear with me. And we're going to do this one here, methyl-directed mismatch repair by McGraw-Hill. I will be sending you links to this in this afternoon's email, so you'll be able to watch it again. Here we go.
Although rare, incorporation of base pair mismatches during DNA replication does occur. There are DNA repair mechanisms that can fix such mistakes. One such mechanism is the methyl-directed mismatch repair system. In Escherichia coli, this repair system can detect a mismatch shortly after DNA replication and repair the newly synthesized strand, thus preserving the base pairing that was present in the original DNA molecule. The system relies on the fact that methylation of a newly synthesized DNA strand does not occur until several minutes after the strand is made. This provides a window of time during which the newly synthesized daughter strand in a DNA double helix can be distinguished from the parental strand, which is already methylated. A sequence of DNA with only one methylated strand is said to be hemimethylated. Actual recognition of a base pair mismatch is the role of the mut-S protein. Mut-S binds to a mismatch site and forms a complex with another protein, mut-L. The formation of this complex recruits mut-H to the region. Mut-H binds to a hemimethylated site. The mut-S, mut-L complex interacts with mut-H by a DNA looping mechanism. Mut-H then makes a cut specifically in the non-methylated strand. An exonuclease digests the non-methylated strand from the cleavage site to just beyond the mismatch site. This leaves a gap in the daughter strand which is filled by DNA polymerase III and then sealed by DNA ligase. The end result is that the mismatch has been replaced with the correct base pair as indicated by the original DNA strand. Although
Okay, folks, so you can see in the video that this is one kind of repair system that, in particular, bacteria rely on, and they make the assumption that an older strand of DNA that has these epigenetic tags, in this case methyl groups, they're assuming that the older version is the correct version of DNA, and that the younger version is probably where the error actually is. So then they snip that out using that enzymatic machinery that the video showed you and fill it back in with the correct complementary base pairs. Do you have questions right now? Okay. Don't worry about the mechanistic details of these things. If I want you to know the mechanistic details of these things, I will put them into your notes, or I will give you some hints that, hey, you need to know about hemimethylated DNA and things like that, okay? But for right now, we're just giving you an overview of things that science has cataloged over the years. Okay. Let's talk. Okay, I'm not going to talk about everything that your book covers. There's just no time to cover it all. But I would like to at least talk about photoactivation repair. Okay, this is something that is covered on figure 12, page 318. Okay. Okay. You know, I mean, everybody knows at this point that you do have to be careful going out into the sunshine. You don't want to get exposed to too much sunshine without some sunscreen on your skin because it may promote the formation of skin cancer down the road. Well, a lot of skin cancer is caused by mutations that are in fact induced by too much solar radiation penetrating the cells of your dermis and epidermis, parts of your skin. Okay. So photoactivation repair involves the use of enzymes to repair thymine dimers that form during ultraviolet radiation exposure. Okay. And so I do have a quick video to share with you on that. Again, we're talking about figure 12 in your textbook. And remember, I also highlighted a specific kind of mutation on figure 8 of your textbook on page 313. So figures 8 and 12 are relevant for this conversation. Okay, gang, here we go with the next video. Thymine dimers, formation and repair. I will send you this link. Here we go.
When cells are exposed to sunlight, radiant energy can damage the DNA. For example, ultraviolet irradiation causes covalent bond formation between adjacent thymines on the same strand of DNA. When DNA is damaged in this way, it cannot be replicated or transcribed. Most cells, however, are able to repair damage to DNA. For example, an enzyme called photolyase can break the covalent bond between the thymine dimers. This enzyme requires visible light, which is used as the energy source for bond cleavage. Hence, this mechanism is referred to as light repair. A second repair mechanism involves an enzyme that excises the damaged segment from a single strand of DNA. This process is termed excision repair or dark repair and does not require visible light. The excised nucleotides are then replaced by DNA polymerase. DNA ligase forms the final phosphodiester bond.
Okay, gang. So, be careful in that these videos make things seem overly simplified. You appreciate that there's a fair amount of complicated biochemistry happening for these processes to work properly. It's just that we're choosing not to get down into the weeds at this moment of time. Okay. Any questions about photoactivation repair?
So light damages the DNA, but then light is the mechanism that fixes it too?
Correct, Hayden. In this particular case, there's an enzyme that's actually activated by being hit by photons of light. So it knows to become active when it gets hit by light, and it actually goes around in the cell scanning for errors. And if it finds them, it repairs them as best it can.
So what if the DNA damage is like deep in your body and it's not accessible to the light?
Right. In such a case, then you could not use the mechanism that I just showed you. You'd have to use a different mechanism. And there are different mechanisms to do that. For example, figure 13 in your textbook, page 318, is an alternative repair mechanism called base excision repair. In such a case, a location deeper in your body would use something like that.
Okay.
Okay. All right. Keep the questions coming. One moment, I have to read a question here from Jose. Okay. Okay. Yes, Jose, no problem to your question. We can take care of that. All right, gang. So let's continue here. The three repair processes that I put into your notes so far, they make the assumption that the mutation in the DNA is on one or the other strands of the double-stranded helix, right? One or the other. In other words, they make the assumption that there's damage to only one strand of the DNA at a time. But what the heck happens if you have a mistake or a damage to both strands of the DNA simultaneously? That's really, really serious and a lot harder to deal with. Okay. So let's see here. Repair processes 1 through 3 are effective when mutations occur on one or the other strand of the double helix. If you have damage to both strands simultaneously, this is much more serious and much more difficult to deal with. Okay, the fact of the matter is that if you have a double-stranded break in the DNA, if both sides of your ladder have been severed for some reason, that can seriously lead to death of the cell. I mean, many cells die because of that very reason. But I'm going to show you two processes that cells can use to sometimes rescue themselves from these horrible, horrible damage events. So let's continue here, and I'll preface this with saying repair mechanisms to breaks in both strands of DNA. Number 4. Okay, I'm going to show you a video on a process that is talked about in your textbook on page 320. On figure 16, it talks about homologous recombination repair of double-stranded breaks. So this is homology-dependent double-strand break repair. Okay, you may remember a few weeks ago during the Thursday extra hour, some of you stayed with me and we stepped through the process of homologous recombination that meiosis will use for crossing over. Okay, that was the main talking point at that moment of time. But I also mentioned, and we also put in your notes, that same process using that holiday junction, if you remember, it kind of looked like a weird plus symbol, the holiday junction, that can also be used to actually repair damaged DNA. And so I'm going to show you a video that emphasizes that at this time. Okay, so, all right. I would like to take note, okay, so this is figure 16 in your textbook, page 321, that this process that I'm going to show you right now also explains, okay, I'll put this in your notes, this process also explains how the molecular details of gene conversion take place, right? Gene conversion was a topic that was talked about in the DNA replication chapter. I didn't really go into it in much detail because we had plenty of other things to talk about. But gene conversion was a vocab word and an answer to one of your exam 2 questions. The idea that when you have damage to the DNA, repairing the DNA can actually lead to a conversion of the allele on one strand of the DNA to the alternative allele. This video is going to show you how that can take place. Okay, so it just emphasizes how different topics and different chapters really are interrelated, and it's kind of hard to talk about everything at one time. You just have to go through enough that you can begin to connect the dots. All right, folks, let me show you this video on homology-dependent double-strand break repair. Okay. Here, that's not it. Okay, here it is. I will send you the video link for this later this afternoon. Here we go.
To see how a cell repairs a double-strand break using DNA homology-dependent double-strand break repair, let's imagine that we have a parental DNA duplex containing the sequence A, B, C, D, E. We then imagine that the duplex is broken by a double-strand break in the C region. To begin the repair process, double-strand breaks in the DNA first undergo nuclease degradation to yield duplexes with 3'-ended single-stranded tails. These tails may be hundreds of base pairs long. In a process mediated by recombination proteins, one of the single-stranded 3'-ended tails from the broken parental duplex, shown in grey, interacts with a homologous duplex, shown in pink, such that the grey single strand invades the pink duplex at a region of homology and forms a region of heteroduplex containing the invading grey single strand and the complementary strand from the pink duplex. The displaced pink strand, which is of the same polarity as the invading grey strand, forms a loop. This loop structure, together with the invading strand, is called a displacement loop, or D-loop. The 3' end of the invading strand then acts as a primer for DNA synthesis. The complementary duplex strand serves as the template. However, this D-loop replication does not form an extended region of heteroduplex between the newly synthesized green strand and the template pink strand. Rather, a small bubble containing the template, a short region of the newly synthesized DNA, and the displaced strand forms and tracks across the pink template strand. This DNA synthesis bubble translocates across the pink template strand until new DNA complementary to the other side of the double-strand break in the grey duplex is synthesized. The replication bubble dissociates, and the newly synthesized strand, shown here in green, is then captured by the 3' single-stranded tail from the other side of the break in the duplex. This capture forms heteroduplex DNA containing the newly synthesized strand, shown in green, and the strand from the broken duplex, shown in grey. The newly captured 3' hydroxy end of the broken duplex then serves as a primer for new DNA replication across the break and extending to the other side of the break. Remaining gaps are then filled by replication and ligation. Note that overall, the gap at a double-strand break where degradation occurred, the C region, has been repaired by new DNA synthesis, the template for which was a duplex homologous to the broken DNA. We see how a DNA double-strand break can be repaired with a more imprecise join in another video, how to repair a double-strand break in DNA by non-homologous end joining.
Okay, gang, so as you can see, pretty involved. Don't worry about the molecular details. That's not what I want you to focus on at this time. I simply want you to be aware of these processes. Okay. In this process, as I'm typing into the Word document at this time, did you notice from the video that if there is a double-stranded break in an area of DNA of one chromosome, then the homologous chromosome can be used to actually synthesize a single strand of nucleic acid that can be used as a bridge to repair the other chromosome in the area where there was in fact that double-stranded break. Okay, folks, question coming through the chat window. Okay, I know these are intense things. I need to ask you, are you with me? Is this making sense? Once again, do you appreciate that in a double-stranded break of a DNA molecule that's part of one chromosome, you can actually use the homologous chromosome in a diploid cell, like a human cell, to repair that double-stranded break by making a fragment of DNA from the undamaged homologous chromosome that can be used as a bridge to seal the damaged version of DNA. Okay, thank you, Morgan, for your feedback. I appreciate that. What can I help you with that may not be making sense? Let me type this down into the notes. So John asked, by using the homologous chromosome, does that erase all of the methylation and customization? It will momentarily, John. Yes, it will momentarily. Assuming that that cell survives, that newly repaired section will potentially be methylated or experience some other epigenetic modifications, but initially in the hours after the repair, yes, that will erase those tags. Okay, so in this scenario, the damage to one DNA molecule of one chromosome can be repaired by using the homologous chromosome to manufacture a bridging molecule to seal the break in the damaged DNA of the damaged chromosome. Okay. So I'm gathering that you seem to be okay with this. I hope so. Folks, here's another question coming to you. If this, what kind of diploid status would repair the damaged DNA in a way that promotes gene/allele conversion? What kind of cell would we be talking about if we were going to get gene conversion? Does that question make sense to you? What type of diploid status would repair the damaged DNA in a way that promotes gene or allele conversion? Do I need to ask that a different way? Okay, I'm hearing a lot of silence, so I'm assuming maybe I need to ask that a different way. So let me ask it maybe more plainly. Would it be a homozygous or a heterozygous circumstance that would promote gene conversion? Please type the answer into the chat window if you know.
Heterozygous.
Okay, thank you very much, Hayden. Absolutely correct. Doesn't it make sense that if you're using a different version of the gene, a different allele, a capital D as opposed to a little d, that when you use the alternative allele to repair a defect in the first chromosome, that you're going to convert that first chromosome to a copy of the template you're using. So that would happen if you're using a heterozygous locus. Okay. Heterozygous would promote gene conversion. Any questions, gang? All right. Okay. So let's move this forward, and we should be able to wrap up the chapter after this next one. There's one more scenario. You know, double-stranded breaks in DNA are really serious, but homology-dependent double-strand break repair can rescue the cell, assuming that you have at least a diploid cell, so there's 2 copies of every chromosome. But what happens if we're talking about chromosomes where there really is no second copy? What if we have a double-strand break repair in a male cell in a Y chromosome, and there's no homologous chromosome available? I mean, the X chromosome is really not homologous to the Y chromosome. What the hell are you going to do then? That's extremely serious. So there is a possibility called non-homologous end joining. And I want to show you a video on that. Again, don't worry too much about the molecular details. Just try to get an overall feel for what the concept is trying to tell you about. Your textbook does not have a graphic on this, and so again, I'm not going to expect you to understand the intimate details. But this is a pretty neat process that we're fortunate enough to have a pretty good video on. This process is alluded to on page 320 of your textbook, okay? And so let me run this video forward so you can see it. Okay. Non-homologous end joining. I will send you this link later today. Here we go.
DNA's role serves high importance, and yet it's vulnerable. Endogenous factors such as replication errors and exogenous factors such as ionizing radiation and X-rays can cause double-strand breaks. Once a double-strand break has formed, within seconds the Q heterodimer recognizes the break and binds to the DNA ends. Q's high affinity for DNA and abundance in the cell allows for its fast binding. It recruits the DNA protein kinase catalytic subunit. This relocates Q inwards so it can access the broken ends. Together, they form a synaptic complex called DNA PK, which bridges the broken ends together. The nuclease Artemis is sometimes recruited to process these ends, removing any damaged or incompatible nucleotides. In general, the process has little to no end processing. However, a small subset of breaks requires it for accurate repair. 2 structurally similar proteins are recruited, XRCC4 and XLF. Their heads contain highly conserved regions, which interact, creating a scaffold that forms a bridge between the DNA strands. The XRCC4 and XLF dimers alternate, forming a long, grooved, superhelical bundle of filaments. I'll leave one here for simplicity. This bundle aligns the ends for ligation. Ligase 4 is recruited in a complex with the tail of XRCC4. It interacts with the 2 broken ends, undergoing a conformational change to encircle the DNA. Ligase 4 modifies the ends, ultimately forming a phosphodiester bond, ligating the ends together. Non-homologous end joining is error-prone, due to its flexibility to directly ligate ends regardless of sequence and without the need of a homologous template strand.
Okay, folks. So in this scenario, this is going to be like a doomsday scenario where the cell does not have access to a homologous chromosome region that it can use to build a template to fill in the missing DNA. In this scenario, you're really going to take the 2 frayed ends of a strand of double-stranded DNA that have separated and use multiple enzymes to glue them back together. Unfortunately, in some ways, that means you're going to potentially lose a series of nucleotides that was in the original DNA and will not be present in the fused DNA that results after you seal the broken ends back together. So that can lead to mutation of a gene in a variety of ways that might make it the case that the gene can't express itself properly anymore, or you make an altered protein than what you originally would. Now, obviously, those are not good outcomes, but they're still better outcomes than having the cell literally die because the DNA of the chromosomes has remained fragmented. Okay, so this is a last ditch repair process that cells use when no homologous DNA is available to repair the break in the DNA. Okay, this process will often lead to mutation of genes. This process will frequently mutate genes, but at least allows the cell to survive. John, you asked a question for the gals. Can the X chromosome use the bar body X chromosome in a homology-dependent repair? Yes, typically it can, John. Okay, and so just taking that a little bit further, John, that's one of the reasons that certain genetic diseases that typically show up in males at a high percentage rate of the population, like muscular dystrophy. Muscular dystrophy is almost exclusively a male disease. Really doesn't show up too very frequently in the gals because there is a second X chromosome that can be used to repair that deficiency or at least complement the defective gene. Okay. All right. Gang, I don't want to belabor a dead horse here. We have many other things to do. So I would like to get on to the next chapter, but before I do you have any questions right now from chapter 11? Okay. All right. Well, gang, let's get on to chapter 8 then. Chapter 8 is about the structure of DNA, okay? DNA organization in chromosomes, not the biochemical structure so much as its organization in the chromosomes. Now, you might wonder why I didn't cover this earlier in the semester, and I feel that the content of this chapter actually makes more sense after you understand things like DNA replication. So I choose to do it after we study DNA replication. Okay, let's see what we can make of this, gang. So chapter 8, page 212, 213 in your textbook. All right, so just some opening points, some things that you already know about. Okay, you know that chromosomes are Mother Nature's biological units to move genetic material around, right? I'm talking about the chromosome, not the chromatin. Chromatin is for expressing the genes, right? Chromatin's for expressing the genes. Chromosome is for literally moving the genetic cargo around from one cell to the next, okay? Okay. While chromatin is used to express genes, chromosomes are used to move genetic cargo around, such as is needed during cell division. Chromosomes are much better at physically moving cargo around than expressing genes. Okay. Okay. So Now, when we talk about life on the planet, there are a variety of different kinds of life on the planet that have chromosomes, right? First, we have viruses, which is extremely relevant right now. Viral chromosomes, they can be made of double or single-stranded DNA or RNA. And typically, the DNA or RNA is associated with a few viral proteins, okay? You see an example of a viral chromosome on figure 1, page 213. And, well, let's see here. Right, something like the influenza virus, right? The influenza virus has a DNA chromosome, right? DNA chromosome. But the HIV and coronavirus that's giving us a hard time right now, and coronavirus both have RNA chromosomes. Okay, so I know some of you haven't had microbiology yet, but that's sort of weird. I mean, cells, when we talk about cellular chromosomes, cell chromosomes are always made of DNA. They are never made of RNA. Never, ever, ever has a cell with an RNA-based chromosome been discovered. I mean, maybe one will be discovered tomorrow, but so far we haven't done that. So viruses are a little peculiar and sometimes don't obey the typical rules that we apply to cells. Okay, when we talk about prokaryotic ribosome, not ribosomes, I'm so used to talking about that, prokaryotic chromosomes, okay, we're talking about chromosomes that are made of double-stranded DNA. That is typically associated with more proteins than a viral chromosome, but fewer proteins than you would typically find with a eukaryotic chromosome. Made of double-stranded DNA associated with more proteins than a viral chromosome, but fewer proteins than a eukaryotic chromosome. Okay. You see an example of a prokaryotic chromosome in figure 3 of page 214. And you can see how all this mess of DNA has spilled out of that E. coli cell. I think I heard a figure a while back ago that the length of the E. coli chromosome that you see in figure 3 of page 214, the length, if you were to measure that length, it would be about 1,500 times the size of the actual length of the cell. I mean, it's insane how big that chromosome is when you unravel it. Okay, so let me mention here some examples of prokaryotic proteins. That associate with the DNA to form the chromosome. Okay, this will not be an exhaustive list in any way, but prokaryotes typically have proteins that are designated HU, and HU stands for histone-like. Okay, if you're familiar with the human body and human anatomy and physiology, and you remember human DNA, well, eukaryotic cells, their DNA is complex with proteins that are called histones. Bacterial chromosomes do not have histone proteins, but they do have proteins that resemble histones. So this is a histone-like protein, which is similar to the eukaryotic H2B protein. And I'm going to put something down in your notes that may not mean too much right now, but hopefully by the end of the lecture, it's going to mean a lot more. I will tell you that the H2 protein, correct my note here, the H2 protein is a protein that induces negative supercoiling in the DNA. And while that may not yet mean anything, it will definitely mean something to you by the end of today's lecture. Okay, areas of the DNA where you have negative supercoiling tend to sort of relax and unzip the DNA enough that you can get some localized pockets of gene expression. Okay, the meaning of the U in the name is actually been lost to scientists. It's not clear why anybody named it that way a long time ago. Okay. Obviously, H is for histone. The U, nobody knows what that's for. Meaning of H is histone. Meaning of U has been historically lost. Okay. And then we have another protein, HNS, which is histone-like nucleoid structuring protein. Histone-like nucleoid structuring protein. A nucleoid refers to a structure of a prokaryotic chromosome. And I'm going to show you a video on this pretty soon, so you'll see what I'm talking about. A nucleoid, not a nucleotide. A nucleoid is a type of structure found within prokaryotic chromosomes. Okay, and the purpose of this protein is to help regulate the process of gene expression. Okay, so that just gives you an idea for a couple of the proteins we typically find associated with prokaryotic chromosomes. Prokaryotic chromosomes are structurally more complex than viral chromosomes, as you might imagine to be the case. And we're going to be talking about that in considerable detail here in about 5 minutes. But just to complete our opening talking points here, we also have eukaryotic chromosomes. Okay, eukaryotic chromosomes. These would be the chromosomes of fungi, algae, protozoa, multicellular animal parasites, animal and plant life on the planet, right? Human cells or your tulips outside. Okay, these are all eukaryotic cells. These chromosomes are made of double-stranded DNA associated with many proteins. Okay, many proteins. And we'll be talking a little bit more about that, if not at the end of today's lecture, then the beginning of next lecture. Okay, so figure 3 in your textbook, page 214, gave you an example of a prokaryotic chromosome. And if you want to look at some examples of eukaryotic chromosomes in terms of the pictures of them, you could look at figure 5 on page 216. That gives you an example of the polytene chromosomes of fruit flies. Figure 6 also shows you that. And the so-called lamp brush chromosomes that kind of have a brush kind of appearance in oocytes, amphibious and oocytes is shown on figure 7. Okay, so kind of a show and tell that you can see that these things are in fact in your textbook. Okay, gang, so that sets the stage for what we're going to be talking about in this chapter. We're going to be talking about chromosome structure, and I am going to emphasize prokaryotic chromosome structure, at least initially, and then later on we will be talking about some we will be talking about some eukaryotic chromosome material, and dabbing our toes into the water for epigenetic modifications. Okay, before we get any further, do you have any questions right now?
So I do have a question regarding viruses in particular. So my understanding of the coronavirus is that it has one of the largest genomes of any RNA viruses that we know of. My question is what the implications of that would be from maybe a pathophysiology standpoint of the larger genome.
Well, it's difficult to predict, Kevin, but obviously the larger the genome, the more information capacity in the viral genetic material, meaning it can probably code for proteins we have not actually seen in Mother Nature yet, or we're not aware of that a typical coronavirus can manufacture. If I can find it this afternoon, I actually read a New York Times article recently. It did a pretty nice write-up about the genetic architecture of what we currently know on the coronavirus, and it does in fact highlight some genes that we don't have any idea what these things are doing. So I would say simply that the larger the genome, the more likely it's coding for molecules we don't currently understand. Okay.
Thanks.
Was there anything else about that that you'd like me to talk about, Kevin?
I was just curious if there were any specific known implications of having a larger genome as far as complexity or I don't know, how serious of an infection it could cause, et cetera.
Well, okay. I would caution you to think about complexity as always meaning larger, because right, there are bacterial genomes which are a lot smaller than a human cell genome, but they still possess different genes in that smaller genome, which allow them to perform unique forms of biochemistry that our own cells can't do. So complexity is not always tied to the size of the genome, although it may in fact be. Okay, if I can dig that if I can find that article, I'll also attach that with today's email. Okay. Anything else? Okay. All right. Yeah. There's so much more I could say about that, but I don't want to get off on a tangent here. Okay. So let's continue. What I want to do is talk to you about topological characteristics of closed circular DNA. Something you probably want to talk to Grandma about. Okay. So first off, okay, your textbook on page 215 introduces you to the idea on figure 4 of page 215, that DNA inside the cell can have different geometries, if you will. Okay, different geometries. So on figure 4, you notice the first geometry is simply to be a linear chain of DNA. The second geometry is to be a circular form of DNA, so-called a relaxed circular DNA, and I'll talk to you about what relaxed means in just a moment. And then we have underwound DNA, and I will also talk to you about what that means in just a moment. And then we have supercoiled DNA. So what we need to go through here is try to understand some basic features of DNA and chromosome structure so that you can understand how the structure actually impacts the physiology of how cells may or may not express their genes. And this is not a trivial topic. This is going to take us the rest of the day here. Okay, so I'm going to probably be bopping back and forth between the Word document and a PowerPoint graphic that I need to use. But okay, first off, let me see here. Let's define this word topological, or I'm going to define a version of the word as topology. Okay. So here's a definition. Topology. Topology is a subdivision of mathematics. Okay, now don't get scared here because we're not going to be heavy math, but it is a subdivision of mathematics that studies the properties of objects when those objects, like DNA is an object, studies the properties of those objects when those objects are deformed, when they are twisted, when they are stretched. Okay, so if you look at figure 4 on page 215, you notice we have the same DNA molecule. It's exactly the same DNA molecule represented 4 different ways. In picture A, it's represented as a linear chain. Picture B is a circle. Picture C is a different circle. And picture D sort of looks like a chain at the end of a tow truck. Okay, so all of these different forms are forms of the same DNA molecule that have a different shape or structure, if you will. So the study of that, of the transitions of one form of DNA to the other, is actually called topology. This is a mathematical subdiscipline that studies, okay, this is a mathematical subdiscipline that studies the properties of objects like DNA, right? All we care about right now is DNA, but it could be, you know, the property of a Chinese yo-yo, could be the property of a piece of toilet paper that you're folding into an origami pterodactyl. Okay, you see what I'm saying here? You can take an object and fold it and manipulate it in different ways. So studies the properties of objects like DNA that are preserved through various deformations, twistings, and stretchings of the object. Okay, so this, you know, this could be a little scary. Don't worry, this is going to be as clear and gentle an introduction to this topic as I can give you. Okay. All right, so let's begin. Okay. First thing, number 1, recall from chapter 6 that the Watson-Crick model of DNA specified that there was a certain number of base pairs for every turn of the double helix. How many base pairs per turn of the helix did Watson and Crick say there was? Please type that into the chat window. Okay, Hayden, wonderful. 6. Okay, I'm sorry, 10. I was thinking about another thing. 10, exactly. You are correct. Recall from chapter 6 that the Watson-Crick model of DNA specified that a double-stranded molecule of DNA contained 10 base pairs, 10 nucleotide pairs of DNA, 10 base pairs of DNA in each turn of the double helix. Okay, that's what we put into your notes when we were covering that chapter. That's what you know, and I'm gratified to see that you are in fact remembering that. That's wonderful. Okay, well, I know speed up to modern times, the more accurate measurements of that base pairs per turn is actually somewhere about 10.4 to 10.5. And we're going to go with 10.5 to make the math I'm going to show you a little bit easier. So modern measurements update the 10 base pairs per turn to about 10.5 base pairs per turn of the double helix. Okay. All right. Please stop me if I'm confusing you. Please stop me if I'm going too fast. Please stop me if you don't understand, because my fear is that if I lose you early in our conversation here, you're going to stay confused the rest of the way, and I don't want that to be the case. So please stop me if you are confused. Okay.
Could you give us a second to finish writing? Just a second.
Thank you. Sure. Okay, are you okay, Hayden?
Yeah.
All right. Okay, so we have a more modern measurement of 10.5 base pairs per turn. That's what we're going to be using today during our overly simplified calculations. Okay, so in just a moment, I'm going to show you a molecule of DNA that is made of 105 base pairs. Okay, so if we start with a DNA molecule that has a length equal to 105 base pairs, if each turn of the helix has 10.5 base pairs, then hopefully you can appreciate that that means we are going to have 10 turns in the DNA molecule, right? 105 divided by 10.5 is 10. Okay, if we start with a DNA molecule that has a length equal to 105 base pairs, if each turn of the helix has 10.5 base pairs of DNA, which current data suggests is correct, this means that that molecule should have 10 turns in the entire molecule. Okay, I will come back to this note if you haven't quite finished it yet. All right, so let me go and show you a graphic at this time. Okay, so here's an image that I'm going to be repeatedly referring to. So I need to know something to see how the video is working on your end of things. Can you tell me what phrase I'm currently circling with my cursor? Can you?
W equals WN equals 1?
Okay, okay, W equals -1, does that make sense?
Okay.
Okay, I just needed to know that you folks can see what I'm highlighting with my cursor, because I'm not sure that the cursor movement is actually recorded in the videos that I'm saving for you that you may be watching after class, which means that sometimes you may not be able to understand what I'm talking about on the video that's recorded. So, all right, so you can see it at least live here. That helps me. Okay. All right, so let's take a look at what we have here, and I'm going to be sending you a boatload of content this afternoon, folks, but hopefully after today's lecture, most of it will make sense and not scare you. Okay, so let's take a look at this. If we start with picture A here, okay, if we start with picture A, right here we have double-stranded linear DNA, 105 base pairs long. That's what we put into your Word document. There are 10.5 base pairs per turn. Okay, that means that a characteristic of this DNA called the twist has in fact 10 turns in it. Okay, so okay, can't ask you to do it since you don't have this at your house yet. But okay, let's take where my cursor is, right under the number 6, and let's literally go around the DNA, counting the number of twists. Remember, a twist is an entire 360-degree rotation of one strand of the DNA. Okay, so I'm just going to move my cursor here. We're starting here at the 6. We come back, we come up 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Folks, do you see that there are in fact 10 turns around this circular molecule of DNA? Does the 10 make sense is what I'm asking. Okay, John, you're okay? Good, okay, good. All right, very good. All right, so let's define twist then. If you understand what twist is, let me just define that for you back in the Word document. Okay. Okay, so twist. Okay, I'm going to define twist two different ways. Twist is the number of turns in a circular molecule of DNA. Number of turns in a circular molecule of DNA. It is also, it can also be expressed differently. It is also the number of 360-degree turns in a single strand of the double helix. And if you don't believe me, do what I just did or tried to do for you when I send this picture to you and you're in your own study time. Pick an arbitrary starting location of that helix, pick one strand of it, and make sure that every 360-degree rotation you count as one, and count the number of those rotations around the entire molecule. You should get 10 for the graphic that I'm going to share with you. Okay? Any questions before we continue? Okay, right now, believe it or not, you are doing some very simple topology. There are some very geeky mathematicians around the world that would be smiling at what you are doing right now. Okay, all right, let's go back to the PowerPoint. All right, so now we know what twist is. All right, now we got to go to a couple related ideas here. A related idea to these turns and twists. Okay, so you notice up here where my cursor is, you notice that the twist has 10 turns in it, right? We proved that to you graphically just a moment ago. Okay, so this DNA that the graphic is showing you is actually linear DNA that they're happening to fold into a circle here. And they're saying, okay, let's go to letter B, where we actually take this linear strand of DNA and covalently link the ends and actually get a full sealed circular DNA molecule. Okay? All right, so a related idea to these turns and twist concepts is that of linking number. Okay, linking number for B is 10, for C is 9, and for D is 9. And I'm going to explain that to you next, okay? So the linking number is the number of times that the two strands of the double helix cross over each other divided by 2. Okay, so let's start here at number 6 again, and we will start counting. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. I don't want to go here because I did that one already. So 20. 20 divided by 2 is in fact 10. Okay, so let's define linking number in your notes. Linking number. Linking number is the number of times the two strands of the double helix cross over each other divided by 2. The number of times the two strands of the double helix cross over each other divided by 2. Okay, so in our example, we counted 20 crossings. You divide by 2, that's your linking number. Okay. All right. So I will be coming back to this if you haven't quite finished your writing on it. Another important property of DNA topology is the writhe. Okay, you notice the writhe here is 0 in letter B. Letter B, the writhe is 0. Letter C, the writhe is 0. And letter D, the writhe is -1. Gang, the rise is the number of supercoils in the DNA molecule, the number of supercoils. Now, it can be a positive number or it can be a negative number depending on the type of supercoil that we're seeing, and I'll talk to you about that in just a moment. But obviously here the rise is -1, so we're saying that this molecule of DNA has one negative supercoil. Okay, let's define that. We'll come back and talk about it even more. Okay, and just so you see that I'm not completely insane. I mean, your book does talk about linking number on page 215, so I'm not giving you too much more than what your book is suggesting is here, although I am giving you a little bit more. So rise equals the number of supercoils. Okay. Now this can be a positive or negative number, depending on the type of supercoil we observe. Okay. All right, so this gives us a chance to talk about supercoil. Okay, now I know supercoiling is something that we've hinted about in other chapters. It was talked about during DNA replication, but we didn't really emphasize it all that much. If we go back here, obviously what you can see between picture C and picture D is in fact that picture D is no longer a nice relaxed circle, right? It's not like you have a rubber band completely sealed and open or in a relaxed state in your hand. We have a situation here where one side of the circle has been pulled over the other side to form almost kind of like a figure 8 kind of scenario. So a supercoil is a structure that's created in the DNA when one double-stranded section of the DNA crosses another double-stranded section of the same DNA molecule. Okay, let's define that. Number 7, a supercoil. This is a structure created in the DNA when one section of the double helix crosses over another section of the double helix within the same DNA molecule. Okay. All right. Any questions right now? Okay. Now, what we have to talk about is this positive versus negative supercoiling business, okay? So, all right. So here's a scenario where I absolutely have to do a little bit of drawing because I can't explain this in words. Okay, so I'm going to go back to the graphic. Okay, so when we talk about positive versus negative supercoiling, okay, so up here, positive versus negative supercoils. Okay, folks, here's the basic rules. Here's the basic rules of how you should interpret this. There are actually rules to this, and while these rules are somewhat arbitrary, they're arbitrary in the sense that mankind has defined these rules. Mother Nature doesn't care what these rules are. Mankind has defined these rules, and because we all want to be speaking the same language, we all have to know these rules to be sure that we're interpreting information the same way. All right. So a positive supercoil is going to be something like this. Okay. Bear with me a moment. Okay, I'm drawing an arrow which represents one strand of the double helix, and then another arrow where there's actually a gap. Okay, so in this scenario, gang, I hope you can come, is it coming out through the screen here that I've drawn two arrows? You tell me, can you see that one arrow is going underneath the other arrow? One arrow is going underneath the other arrow. Okay, all right, very good. I need you to appreciate that, okay? All right, so this is the rule for interpreting a positive supercoil. Okay, and we flip that circumstance to get a negative supercoil. So the negative supercoil rule is this, followed by this. This is the negative supercoil rule. All right. So, so what I would like you to do now with these rules is ask yourself this area here. If you were to interpret this area that I'm sort of highlighting with my cursor, and let me see here, I'll put an orangish yellow in that area. Okay, what I need you to do sitting at home is look at this area and interpret it according to these rules. Please tell me, according to these rules, whether you think this is a positive or negative supercoil. Go ahead and type your answer into the chat window. Okay, John says it's positive. Please type in your answer into the chat window. Okay. Kevin thinks it's positive. Okay, good. All right. Okay, I'll wait for, all right, okay. Folks, basically what I need to know is that you are processing this information. Don't just copy somebody else's idea down. Make sure you're looking at it and you're reasoning it through in your own brain. Wonderful. All of you have said positive, but the handout says it should be negative, right? So there's something that we're missing, and I did that intentionally to you. These rules are correct, and the answer on the picture in front of you is also correct. This is not a typo, and each of you has in fact interpreted these rules correctly, but I didn't give you all the necessary information yet. So let me ask you, we are technically at the end of our time together. I really, really want to continue this conversation because I'm hoping to start chapter 12 next Tuesday. Let me type in it and a question to the chat window. Can you continue online with me at 2 p.m.? Okay, all right, okay. I mean, it doesn't make much sense for me to sit here and talk to myself. So if you really can't stay online, then it doesn't make sense for me to do this. But okay, it seems like most of you can. Gang, I need a few minutes to check on my little boy. So let's take about a 12-minute break. We will be back at 2 p.m. sharp, and we will continue this conversation so we can get through chapter 8 and be sure we don't get too far behind this semester. So thank you for your willingness to stay on. Let's get back in 12 minutes, and we will continue. So I'll see you in 12 minutes. Okay, gang, welcome back. Are you ready to continue? Okay, great. Thanks for sticking with me. All right, so we were talking about the supercoiling business, and we had defined these rules to interpret what a positive and negative supercoil was. You all interpreted this section highlighted in the yellow region and said that this is a positive supercoil. The logic of what you've applied is absolutely correct, but it's still a wrong answer because there's a piece of information I intentionally left out. Here it is, gang. Okay, between prokaryotic cells and eukaryotic cells, DNA adopts two different conformations, two different kinds of shapes, if you will. And knowing what shape you're looking at from what kind of cell you're looking at helps you define the proper viewing perspective of how to interpret the rules. So the reason the logic of what you answered was incorrect is because we haven't told you about how to determine the proper viewing perspective to interpret the rules. So let me type these out in the Word notebook, then we'll come back. Okay. So positive versus negative supercoiling here. To apply the rules of interpretation, which remember, go to the PowerPoint figure that I showed you. Okay. You must adopt the proper viewing perspective for the DNA you are observing. Okay, so key here is the viewing perspective. And again, this is entirely arbitrary in the sense that mankind created this, nature didn't create it. Mankind has created this, but again, for us all to be talking about the same thing when we have a conversation in genetics, we all have to appreciate the same thing. So, number one here, okay, in prokaryotic cells that have circular DNA. Now remember, prokaryotes have circular chromosomes. They also typically have circular plasmids, if you remember that lesson from microbiology. So this is going to be true of chromosomal DNA in bacteria. It's also going to be true of plasmid DNA. The prokaryotic cells that have circular DNA, the DNA is intertwined. Okay, I think that's the best word I can use. It's intertwined, forming linked figure eights. Okay, in prokaryotic cells that have circular DNA, the DNA is intertwined, forming linked figure eights. Okay, I'll give you a moment to jot that down, and I'll talk more about it. Okay, so just for a moment, go back to the window here, but go back to the graphic. What I'm saying is, if you look at the cursor on the screen, right, I'm drawing out a figure eight here. And in fact, when we talk about long chromosomal DNA inside bacteria, you can have something that, you know, kind of looks like this. Figure eight, the bottom of the top one is now the top of the next one, right? A chain of figure eights that are intertwined like this. This is typically the conformation and shape you will find for circular DNA inside prokaryotic cells. And you can see in the example of the handout I've got that this is clearly a figure eight perspective. Okay? So we'll come back to that. Back to the notes. Okay? this type of structure, this intertwined linked figure 8 structure, is actually given a name by scientists. It's called plectonemic DNA. This type of figure 8 structure is referred to by topologists as plectonemic. Okay, nice fancy vocab word there. How about that? Okay, plectonemic. Okay. And here's the kicker here. For plectonemic DNA, the viewing perspective is always perpendicular to the long axis of the molecule. Okay, for plectonemic DNA, the viewing perspective is always perpendicular to the long axis of the molecule. Extremely important to do this interpretation properly. Okay, for plectonemic DNA, the viewing perspective is always perpendicular to the long axis of the molecule. Okay, I'll give you a second to chat that down. Okay, we'll come back to that. Let's go to our graphic here. Okay, now we take a look here. You can appreciate that on our drawing here, right, we're going to have this axis, which I wish I could draw straight. That's the long axis, right? And here's the short axis. And our definition of the viewing perspective for plectonemic DNA is that you always view the DNA perpendicular to the long axis. So, if you understand what I'm saying, I need you to view this area in yellow from a perpendicular axis to the long axis, and I need you to reinterpret the question with your new understanding of a perpendicular viewing perspective. Apply these same rules and tell me whether this supercoil is positive or negative. So imagine you had this paper, imagine you had this graphic on a piece of paper in your hands and you rotated the page 90 degrees so that you were looking this way, where I'm moving my cursor. You're looking this way. And now reinterpret the rules based on this viewing perspective. So with that in mind, take a moment and honestly force your brain to think, is this a positive or negative supercoil? Okay, Kevin. Okay, Morgan. Okay, very good. Okay, good. All right, folks, that's what I need you to appreciate. Okay, Hayden. All right. So in other words, Hayden, rather than looking at the picture as it's currently drawn, imagine that it was drawn like this. If I can get this to work for me here. Okay, imagine that rather than having the figure 8 you currently have, you had this figure 8. Okay? So that's basically what you're doing when you rotate this 90 degrees. Folks, how can we help Hayden understand this better?
But can't you flip it either way?
Can't you flip it either way?
So if I turned it to look like how you drew, if I turned it one way, it would be positive. If I turn it the other way, it would be negative.
No, which way you turn it doesn't matter. So I challenge you, go ahead and answer the question starting from this viewing perspective here, which I'll define as A. So look this way, and then imagine you looked on the other side, that way, are you going to come up with the same answer?
Yeah, I see it now.
Okay, right? All right. So, and no, that's totally okay. This is not the most obvious thing the first time around, so it's okay to spin your tires a little bit. There's nothing wrong with that. Okay, folks, are you feeling okay at this moment? All right. Okay. All right. Again, you know, after class here, I can stick around and try to help you if you're still struggling with certain things. All right, so let's go back and finish our notes here on the notepad here. Okay. So now we have solved the mystery of why on the PowerPoint graphic that I've been showing you, the supercoil is actually negative instead of positive. It all has to do with interpreting the proper viewing perspective to apply the rules. Okay. And just for completion of your notes, in eukaryotic cells that have linear chromosomes, so linear DNA, the DNA is looped around the histone proteins forming a structure that looks like a bunch of linked beads. Forming a structure that looks like linked beads. Okay. The best analogy I can give you on that without drawing it out is, do you folks, I know you do, but you might give me a hard time about it. You do remember what an old-time telephone looked like that had those curly cords connected to the handset. You know what I'm saying? Right? Nobody uses that anymore because everything's wireless. But the old style telephones that had a handset connected to the receiver with a curled cord, that's kind of what the DNA looks like in the linear chromosomes of eukaryotic cells. The old style telephone cords, okay? So in other words, in a eukaryotic cell, rather than the figure 8, it kind of more looks like this. Okay. All right, so going back to the notes, this form of DNA is referred to as solenoidal. It is referred to by topologists as solenoidal. Okay, you'll be getting all these notes I will send to you through the email this afternoon. For solenoidal DNA, the viewing perspective is always parallel to the long axis of the molecule. Okay, so you can see how that would be different. For solenoidal DNA, the viewing perspective is always parallel, not perpendicular, parallel to the long axis of the molecule. Okay. We're doing pretty good here, gang, with time considerations. Okay. Any questions at this moment? Okay. So I'm going to switch to the picture again and point out a couple other things. Okay. So a couple things, I'll be sending you this picture and you have the notes we've taken this afternoon. A couple things I want to point out here that are part of the puzzle of understanding chromosome structure. Okay, so now you know about linking number, you know about twist, you know about rive. A couple other things that I want to point out. Notice that in pictures B and C, there are no supercoils, right? B does not have any supercoils. C does not have any supercoils, okay? And in each respective case, the linking number equals the twist, right? 10 and 10, or 9 and 9. So what I'm saying is that when you have a piece of circular DNA without supercoils, the linking number and the twist are going to be the same. But when you inject a supercoil of some kind, where one region overlaps another region, all of a sudden that relationship breaks down so that the linking number is no longer the same as the twist. Okay, let's put that down into your notes. Okay, let's put that down as number 8. Other observations. Okay. As long as circular DNA does not have supercoils, the linking number will equal the twist. Okay. Now, let's continue. Something else to point out. Going back to the picture here. Notice in the picture C here that I'm circling right now with my cursor. Notice that picture C has this word strained in it. Okay. Notice how B has the word relaxed in it. Okay. Folks, in terms of DNA topology, the natural relaxed conformation of the double helix is that conformation which basically Watson and Crick figured out a long time ago, which is to say that each turn of the helix contains 10.5 base pairs of DNA. So if you have relaxed DNA, you can assume that there are 10.5 base pairs per turn. But if you have strained DNA, you notice that the base pairs per turn changes. Now, in this particular case that we're showing you, it happens to go up, but that may not always be the case. It can actually go down, okay? So strain tells you that the number of base pairs per turn is going to change. Okay. So before I go back to the notes, do you see what I'm pointing out at this moment? Are you okay? All right. And since before I go back, take a look at this, gang. Take a look that when you supercoil, right? When you supercoil, look what the strain circle did. It went from base pairs per turn to 11.67. Look what happened to the base pairs per turn. It went back down. What I'm suggesting to you, at least in the case that's being presented on the screen, is that in this scenario, the presence of the supercoil actually relaxed the DNA because the base pairs per turn went down. Okay, so this arrow here, oh boy, I wish this would behave itself better. This double arrow here in the middle of the screen, you see it's a double-headed arrow. As we go from C to D, we are actually relaxing the DNA so that the base pairs per turn goes down, and that is consistent with a negative rise, a negative supercoil. Okay. All right, let me see if I can put some of this into words in your notes. Okay, so at this point, I'm going to refer to the different pictures in the handout and assume that when you read these notes in the future, you know what handout I'm talking about. It's the one on the PowerPoint screen today and the one that I will email you this afternoon. Notice how the picture C is described as strained. Okay, it's not described as copyrighted, damn it. Okay, picture C is described as strained. Okay, in this picture, the linking number and twist are lower, but the number of base pairs per turn has increased relative to picture B. Okay? Notice how the picture C is described as strained. In this picture, the linking number and twist are lower, but the number of base pairs per turn has increased relative to picture B. Okay? This thing that I'm pointing out is an example of underwound DNA. This means picture C is an example of underwound DNA. Picture C is an example of underwound DNA. Okay, let's pick that apart a little bit. Okay. Let's think about that. Because the size of the DNA molecule has not changed, meaning it is still 105 base pairs long, and there are fewer turns in the molecule. This means that each turn must have more base pairs per turn. This means that each turn must have more base pairs per turn. Okay, maybe read that and say it over to yourself a couple times. Because of the size of the DNA molecule in the picture we're talking about has not changed. It's still 105 base pairs long, and there are fewer turns in the molecule. This means that each turn must have more base pairs per turn. Does that make logical sense? Okay. All right. All right. I'm glad to see we get some yeses here. Okay. All right. So let me summarize some things this way. Okay. Underwound. Okay. Underwound DNA leads to fewer turns, meaning it has a lower twist. And the implication of that is that the base pairs per turn have to go up. You can also have the opposite scenario where you have overwound DNA. Okay? Overwound DNA, you have more turns, meaning the twist is higher. So you get a higher twist. I know this is not everybody's cup of tea, so bear with me. We're getting close to the end. Okay? And so that means the base pairs per turn actually go down. Okay? Continuing with notes that I need to give you so that you can appreciate things moving forward. Okay? Underwound DNA promotes the formation of negative supercoils. Underwound DNA promotes the formation of negative supercoils. Okay? Think about that. Read that. Think about that. Underwound DNA promotes the formation of negative supercoils. Let's think about that here at the picture. We know picture D is a negative supercoil. We proved that to ourselves about 5-10 minutes ago. This is a negative supercoil. And we have a scenario here where, in fact, the twist has gone down and the base pairs per turn has gone up. Okay, so the fact that we have an underwound, this molecule C that you see in picture C here, this is an example of what underwound DNA looks like. Okay, let me write that on the screen. Okay. This is underwound DNA. And because it's underwound, and by the definitions of underwound that I just put into your notes, it makes sense that this, as an underwound molecule, should induce the formation of a negative supercoil. Okay. So let me finish the notes for you. Underwound DNA promotes the formation of negative supercoils, while overwound DNA promotes the formation of positive supercoils. Okay. All right. We're almost going to hit the bottom of this thing, and my next thing that I have to do is try to get you to see how this connects to the physiology of the genetic processes happening inside cells. So before we go any further, I know we've got some, some new terminology and things, overwound, underwound, all this kind of good stuff. Do you have any questions? It may be a scenario where you have to sit down with your own study time in the future and go over this again and make sure you appreciate the logic of things. Okay, so number 9. What is the biological relevance / significance to all this? Well, it's really profound, although I'm not going to make it terribly complicated right now. But if you're following along and you get this, okay, what I'm trying to tell you is that positive supercoiling, okay, positive supercoiling, which is not the picture I've shown today, positive supercoiling tightens DNA. It coils it tighter, okay? It tightens the structure of the DNA up, meaning that the number of turns is up, okay? Number of turns is up. And think about it. If we tighten the DNA because of the positive supercoils, this makes the structure of DNA more compact. And if we make the structure of DNA more compact, it's going to be harder for any enzyme that wants to decode the information of the DNA to do that, which means that positive supercoils, by compacting the structure of the DNA, make it a lot harder to express genes. All right, so let me slow down so you can write this into your notes. Positive supercoiling tightens the DNA. The number of turns goes up. This makes the structure of the DNA more compact, which makes it harder to express the genes because the enzymes needed to decode the DNA cannot access the DNA. Okay. Negative supercoiling loosens the DNA. The number of turns goes down. And this opens up the localized structure of the DNA within a chromosome, making it easier to express the genes. Okay. All right. Let me finish this out, gang, so we don't go over to too much time here. So if you appreciate these statements, positive supercoils compact the DNA, making it easier to turn gene expression off. And negative supercoils loosen the DNA so that the enzymes can access the DNA code. It makes it easier to express genes. Okay. Now the enzymes that create and destroy the supercoils, do you know what they're called? Question through the chat window. What are the names? What is the name? Okay, I couldn't even get the darn question out. Good job, John. Okay. Topoisomerases, that's what I'm after. Topoisomerases refer to a whole class, family, category of enzymes that collectively have the ability to create and destroy supercoils. Enzymes that create and destroy supercoils are topoisomerases. Okay? Topoisomerases are involved in DNA replication. They are involved in transcription. They are involved in the structure, the packing structure of changing chromatin into chromosome and vice versa. Okay. Topoisomerases are involved in DNA replication, transcription, and in converting between chromatin and chromosome states for the genetic material. Okay. Now, one last thing, and then I want to show you a quick little video. All right. Okay, so this is all nice. Okay, Dr. Allen's great. Okay, but give me an example here. Okay, so for example, okay, you may recall during our time talking about DNA replication that there was this enzyme that had to unzip the helix called DNA helicase. Well, when DNA helicase moves through and begins to unzip the DNA that is upstream of the advancing helicase enzyme starts to form positive supercoils, which means that structure upstream of the helicase is getting more compact. And it's going to get harder and harder for the helicase to unzip that section, which means the actual movement of the helicase creates a structure which is going to slow down and even sometimes block DNA replication. But fortunately, Mother Nature is very inventive. So Mother Nature created a version of topoisomerase which will relax the positive supercoils and turn them into negative supercoils. Do you know the name of that enzyme? Let me see if I can answer the question. Let me see if I can type the question before you answer it. What is the name of the type of topoisomerase that relaxes supercoiling ahead of the DNA helicase. Ah, I got it in before you answered. What is it, gang?
Is it gyrase?
Okay, good job, Hayden. It's gyrase. That's right. You have it. Okay, good. Okay, so for example here, during DNA replication, helicase creates, let me back up, the motion of helicase creates positive supercoils ahead of the replication fork. This could potentially disrupt replication. To avoid this, Mother Nature created DNA gyrases, which are a type of topoisomerase, to convert the positive supercoils to negative supercoils. By creating the negative supercoils, we actually relax the DNA upstream of the advancing helicase, making it so that the enzymes can actually continue to do their job and prevent the structure of the DNA from getting so tight and bound up that we stop the process. This relaxes the DNA enough to allow DNA replication to continue unhindered. Unhindered, yes. Okay. All right, gang. So what I'd like to do now is show you about a 4 or 5 minute video, and assuming that you haven't passed out by the end of it, if you're okay with that, then we'll wrap up for this afternoon after the video. Okay? Do you have any questions at this time? Okay. Folks, I'm going to go to this video, and I will send you a link for it later this afternoon, and you'll be able to see it on your own time here. But it should give you a very graphical representation of some of the main ideas that I've been trying to tell you for the past hour or so. So here we go. Okay, here we go. It's called Supercoiling by W.W. Norton and Company. And that's the people that created the video. Here we go, gang. Let's watch this. It's just 6 minutes. Please try to stay focused with the idea of everything we've talked about over the past hour. It will help solidify your understanding. Here we go.
In a remarkable experiment, a cell of E. coli was lysed, releasing its chromosome for electron microscopy. What spewed out of this single cell was a strand of DNA 1,500 times longer than E. coli itself. How could this enormous molecule fit into a single cell? Inside a bacterial cell, the DNA is much more tidy. The DNA is compacted into a structure called a nucleoid. A nucleoid consists of DNA arranged in tightly wound loops, with the boundaries of the loop domains defined by histone-like anchoring proteins. The DNA in the loops is supercoiled. That is, the double helix of DNA coils upon itself, creating a tighter, more compact structure. If one of the strands of a double helix is cut, the DNA loses its supercoils as the tension dissipates. However, the other domains remain supercoiled because they are constrained at their bases by the anchoring proteins, which prevent rotation. To understand supercoiling, consider a completely relaxed circular piece of DNA. An unstressed DNA molecule naturally forms a helix with about 10 base pairs per turn. Now consider cutting a strand, unwinding a turn, and then resealing the DNA. The DNA is now underwound and has one too few helical turns. The DNA will naturally attempt to return to the same number of turns as before, but considering the DNA has been resealed in this underwound form, it must now create a supercoil in the opposite direction in order to relieve the stress. Negative supercoils, such as this one, relieve the torsional strain of underwound DNA, while positive supercoils relieve the strain from overwound DNA. This circular double-stranded DNA has 5 negative supercoils. If the supercoils were removed, the DNA would be revealed to be underwound by about 5 turns. The torsional stress in the underwound DNA is relieved by rewinding and at the same time creating negative supercoils. How does DNA achieve a supercoiled state? A bacterial cell produces enzymes that can twist DNA into supercoils and relieve supercoils. Enzymes that change DNA supercoiling are called topoisomerases because they change the topology of DNA. Type 1 topoisomerases cleave only one strand of a double helix, while type 2 enzymes cleave both strands. Type 1 enzymes are generally used to relieve or unwind supercoils, while type 2 enzymes use energy to add supercoils. Topoisomerase 1 binds to DNA and opens up the 2 strands, loosening the double helix. This loss of a turn in the double helix allows the DNA to convert from 5 supercoils to 4. Topoisomerase 1 cleaves one strand of a double helix, holds on to both ends, and passes the other intact strand through the break, after which it religates the strand. The topoisomerase comes off the DNA, leaving the circular molecule with 4 supercoils instead of 5. An example of a type 2 topoisomerase is DNA gyrase, whose function is to introduce negative supercoils in double-stranded DNA, rather than to remove them. The active gyrase complex is a tetramer composed of 2 gyre A and 2 gyre B proteins. First, gyre B grabs one section of the double-stranded DNA. Then gyre A introduces a double-strand break in the DNA and becomes covalently attached to the breaks as it holds the 2 ends apart. Adding a negative supercoil to DNA requires energy, which is gained through ATP hydrolysis. Gyre A, which is an ATPase, uses the energy of ATP hydrolysis to pass the intact double-stranded section through the double-strand break. Gyre B then rejoins the cleaved DNA and opens at the other end to release the newly sealed strand. The DNA molecule now contains 1 negative supercoil. To maintain proper DNA supercoiling levels, a cell must delicately balance the activities of the 2 types of topoisomerases. The nucleoids of bacteria and most archaea, as well as the nuclear DNA of eukaryotes, are kept negatively supercoiled. Because the DNA is underwound, the 2 strands of negatively supercoiled DNA are easier to separate than positively supercoiled DNA, which is important for transcription enzymes like RNA polymerase that must separate strands of DNA to make RNA. Some archaeal species living in acid at high temperature have a challenge of keeping their DNA from denaturing, that is, separating into single-stranded DNA. To solve this problem, their DNA is overwound, resulting in positive supercoils. It is proposed that the extra turns in the DNA tighten the coil, which would require more energy in the form of heat to separate the strands.
Okay, gang. So hopefully most of what that video was telling you made some better sense because of how we've walked through this over the past hour. And you can see that Mother Nature does some rather interesting things, particularly those cells at the end of the video there where, because of the excess heat energy they typically live in, their genome is typically held in a positive supercoiled state to prevent the DNA from falling apart. And also, did you take note to how the gyrase, particularly the type 2 topoisomerase molecule, really consumed a lot of energy in order to do its work, which is to say, that's why processes like DNA replication that are going to require topoisomerase to do something are extremely energy intensive. It takes a lot of energy to reproduce a genome. Okay. Gang, what questions do you have? Are you okay at this time? Is there anything we need to talk about before we close up shop for the day? Okay, I feel pretty good about things, and I thank you for being willing to hang out a little bit longer than normal. What this will allow me to do is to wrap up chapter 8 in maybe 20 or 30 minutes on Tuesday so that I can get started on chapter 12, right? We're really going to be moving at a brisk pace here. So, okay, thanks everybody. Take care of yourself, stay safe. Remember, literature review due next Tuesday. I'll stick online here a little bit. If anybody needs any more personal attention, I'll be happy to help you. So until then, have a good weekend, gang, and be careful out there. See you later.